Hi, I'm Fairlie, and I'll be showing you my poster for analyzing the ENCODE 4 long-read RNA-seq dataset, which reveals distinct classes of isoform diversity. So alternative isoforms can arise from multiple processes, the first of which is more canonically known, which is from the process of alternative splicing, which basically leads to the inclusion or exclusion of different exons in the final messenger RNA, which can then, in the case of protein coding genes, lead to differential protein domains that are included and therefore different functions of the protein. However, alternative isoforms can also differ at the ends of the transcript, so at the 3' end and the 5' end. In the case of protein coding transcripts, if the coding sequence is in either the first or the terminal exon, and this is altered by which terminal exon you choose, this can confer a difference in the protein that is encoded for. But there are also regulatory regions in the 5' and 3' UTRs of transcripts which can predispose the isoforms to post-transcriptional regulation. Particularly canonical example of this is the 3' UTR being used to bind microRNAs or RNA binding proteins, which can predispose it to some specific post-transcriptional regulatory fate. For the purposes of this study, we'll be using long-read RNA sequencing data, which is a technique which allows us to sequence through the entire transcript molecule, which gives us all the exon structure of the molecule as well as the 5' and 3' ends used in the molecule. And the data I'll be showing you today is the data that my lab generated for ENCODE, which is all of the PacBio human long-read RNA sequencing data. So altogether, we have 51 libraries for 10 unique tissues and 87 libraries for 40 unique cell lines, with over 200 million full-length reads. So this is a really rich data source to be able to explore the human transcriptome. Just a brief overview of our processing. We first map our full-length non-chimeric PacBio reads using Minimap2. We then correct common long-read sequencing artifacts using transcript clean. We then annotate each read to the transcript that it came from and identify novel transcripts as well as filter them. Then we use a new tool that my lab has developed called LAPA, which performs 5' and 3' end calling from each long read and also filters these new 5' and 3' ends based on external validation from assays such as CAGE or poly-A-seq. And then finally, we use another new tool from my lab called Cerberus, which integrates multiple data sources which support the existence of different exon structures of transcripts, so different annotations, as well as supports the existence of different ends of transcripts. So, atlases of promoters or poly-A sites, as well as transcriptional assays such as PAS-seq and CAGE again. So I'm also going to introduce an interesting way that we're referring to each isoform of a gene. So let's imagine that we have this case here where we have these 3 isoforms for a gene. Well, we've kind of boiled it down to each isoform in a gene can be characterized by the TSS that's used in the isoform, the intron chain that's used in the isoform, and the TES that's used. And in the cases where a gene has more than one isoform that's characterized by differences at one of these 3 features, we can kind of add that, like increment the number of the feature that we're looking at and add that to kind of our catalog of transcriptional events that can happen in that gene. So in this case here, we're going to call this gene 111. Because it uses the first TSS that we see, that we've cataloged, the first intron chain that we've cataloged, and the first TES that we've cataloged. In the case of this transcript, we've now added a new intron chain and a new TES, so we'll call it 122. And for this last transcript where we've just added a new TSS, we'll call it 221. We can also refer to groups of transcripts that share characteristics in this space using this syntax here. Where this 1 refers to all those transcripts that use that TSS. And this also enables us to, be able to compare transcripts based solely on their name. So we can see here that these 2 transcripts share the same TSS and these 2 transcripts share the same intron chain. So using this naming strategy, we can just go ahead and look at the transcripts that belong to a specific gene, in this case, MEF2C, which is a transcription factor. And I just want to show an interesting example of cell type specific or cell line specific isoform usage here. So here what you're seeing in this heat map is the relative isoform use of each isoform in this gene compared to the total gene. So in the cases where you see 100 here, this means that this isoform is the only isoform that's being expressed in these three cell lines. And interestingly, it lacks a functional domain, which might lead to it being less functional in its expressed cell lines. So I'll also introduce something to kind of follow up on the idea of referring to transcripts using these isoform triplets, as I should say, basically by using gene triplets instead. So instead of referring to the specific set of features that define each isoform, the gene triplets now summarize the diversity of the features that define all of the isoforms within the gene. So in this case here, we have a gene which has two different intron chains, so we'll call it 121, or its gene triplet will be 121. This one will be 112 because it has two different TESs, 421 because it has four different TSSs and two different intron chains, and so on and so forth. And we can plot the values of these gene triplets on this simplex here, where essentially the genes based on their primary source of transcriptional diversity will fall into one of these sectors of the simplex. So a gene such as this blue one here, for example, that has a lot of different transcription start sites might fall into this category. In the center of the simplex, we have two different fates for a gene, the first of which are genes which only express one isoform, but anything else that falls into the center of the simplex will simply just be called a mixed gene. So just a broad overview of what we see in terms of the transcriptional diversity of our data versus GenCode. We can see that we see a lot more transcriptional diversity at the 5' and 3' ends of our transcripts. And this is in no small part a consequence of us actually paying attention to the 5' and 3' ends of the transcripts. So by using our long reads to call new ends of transcripts, we are contributing to us having more transcriptional diversity at the 5' and 3' end in general. So we can also plot the triplet values for each of these genes in our simplex on a sample by sample basis. So in this case, the triplet is computed from the set of isoforms that are expressed in each sample. And so what you're seeing here on the left are the triplets plotted for each sample in col3A1 for all of the expressed isoforms. But on the right, what you're seeing is it subset even further just for the major isoforms of col3A1. And here we're defining the major isoforms as the set of isoforms that are responsible for at least 90% of that gene's expression. So it's an interesting thing to note that col3A1 seems to shift from having a lot of internal splicing, which is actually largely driven by the expression of a lot of different lowly expressed minor isoforms that exhibit internal splicing. So we're shifting from up here to down here, where now the major isoforms of col3A1 exhibit more simple behavior or tending towards TES diverse. We can also compare the behavior of different genes between species. So here what you're seeing on the left are just the samples which kind of match roughly the samples that we have in mouse as well. And you'll notice that they tend to be in the middle of the simplex, as well as one up here. But in mouse, all of the samples that this gene is expressed in are in this splicing high portion of the simplex. And as a broad overview of what we see that's conserved between the shared genes in human and mouse, around 42% of the sectors of these genes are conserved. So there's going to be some interesting stuff that comes out of this in the future. Speaking of which, in the future, I'm working on adding the plotting code to generate those triangles that you saw today to Cerberus. I'm also interested in coming up with a metric to represent genes in the simplex based on their relative expression values of their constituent isoforms. So that would be an alternative to the major isoforms plot that you saw. I'm also going to characterize the functional potential of alternative isoforms, including their protein coding potential and their microRNA binding site susceptibility. And then similarly to what you just saw in the interspecies comparison, we're also interested in seeing how genes change in the simplex between other biological contexts. So between samples or between our all and major isoform set. And with that, I'd like to thank my large group of collaborators, and I'd like to thank you for listening.